I've just been told I have a caller, and my caller's name is Postnet. Postnet, are you there, darling? Okay, he's still coming onto the line. All right. He would like this question. It's question 1.4, and we're going to be going through this question. All right. Postnet, are you there?
Yes.
How are you, my boy?
I'm fine.
Where are you calling from?
From Limpopo.
From Limpopo. We've had all over the country tonight. We've had one from Cape Town. We've had Limpopo. We've had Mpumalanga. We've had Durban. And you know what? I'm going to be visiting you in December because I'm coming on holiday in Limpopo.
Okay.
Yeah, so you must be ready. All right. Okay,
Yes.
Let's look at question 1.4. The table below shows the DNA. So let's underline. You always underline. You never ever go through a question without a pen in your hand, okay? So
Yes.
DNA-based triplets that code for different amino acids. Now, before you even think of answering this question, Postnet, the first thing you are going to do is this, and promise me, this is what you're going to do. You're going to say that A always combines with T, and that G always combines with C. All right.
Yes.
Now, also remember that in your RNA, there is no T, only U. And what I mean here is adenine will always combine with thymine, and guanine will always combine with cytosine. All right? Always. Yes. Okay? Your RNA, there is never any thymine in RNA. There is only uracil. Now, if you've written this next to any question that you are dealing with DNA, you cannot confuse it. And you will get 100% every time. All right?
Yes.
Do you understand this, Mahan?
Yes.
Okay, fine. So let's have a look. They give us a whole bunch of amino acids, and they give us a whole bunch of base triplets in the DNA template. So these are codons. They code on to it. So for leucine, we've got GAA, which is guanine, adenine, and adenine. You've got histidine, we've got guanine, thymine, adenine. Lysine, we have thymine, 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 and so on and so on. So they're telling you to make a protein, you are going to use all these different little amino acids, and amino acids group together in a chain, and then they start joining each other to form a protein. A protein is a macromolecule. It's a big molecule made up of building blocks or monomers called amino acids. All right, so the following is a part of the sequence of an amino acid. So it's a piece. It tells you it's a part of the amino acid that form a particular protein molecule. So here we go. And they say we got ALA, which is alanine, his, which is histidine. We got TRP, and we go down here, which is tryptophan. And we have lyse, which is lysine. All right. It says, name the process by which messenger RNA is formed from a DNA template. Now we're going to make that messenger RNA first, and it's made from the DNA. That is transcription. Okay, it's transcription. All right, you are transcribing. You are duplicating what is there. Okay, so that's going to be transcription. How many messenger RNA codons would be involved in forming the portion of the protein shown here? That is going to be, come on, 1, 2, 3, 4, 5. Yes, so we're going to have a codon each time because your base triplet is a codon, and there are going to be 5. Then we say, write down the sequence of the first 3 messenger RNA codons from left to right for this portion of the protein. Okay, so what they want you to do here is to write down the first 3 messenger codons. So the first three would be alanine, histidine, and tryptophan. So let's just move this up just a touch. And we're going to say, so the sequence is going to be, the first one is alanine, which is going to be CGA. But remember, that is what we are going to see. This is the base triplet in the DNA. The messenger RNA is going to be the opposite. So we're going to just write those in here just so that you can see what I'm talking about. And then we'll look at what the codons are. So the codon for GAA, remember I said you must always write this on the side. And if we're dealing with DNA, with the messenger RNA, all right, or any RNA, there is no thymine, it's always a uracil. So wherever you put a T, instead you're going to put a U. So if it's GAA, the opposite of G would be C, and the opposite of A and A would be T, but we can't use T, so it would be U and U. Do you understand this? Postnet,
Yes.
do you understand this, hun?
Yes, I understand.
Okay, this one here, you do this one. What am I going to have? What's the opposite of G? It's going to be C.
C.
And the opposite of T is A. And the opposite of A?
T.
No, it's in the RNA. So that's going to be a U. Here we're doing the RNA. All right.
Okay. Yes.
So it's going to be the opposite. And there's no thymine. There's always uracil in RNA. So
Okay.
TTT is going to be?
AAA.
There we go. You've got it. Do you understand?
Yes.
Okay. GGG?
CCC.
There we go. CGA?
GCU.
GCU. Well done. And ACC?
UGG.
There we go. And AAA?
UUU.
And CCT? So, sounds like we're talking a different language here, hey? There we go. You got it. Do you understand
Yes.
now? All right.
Yes,
That's
I
what,
understand.
always remember, adenine to thymine, guanine to cytosine. When it's RNA,
Okay.
there is no thymine, there is only uracil. All right. Will you
Yes.
remember? Okay, so here,
Yes.
if we look at question 1.4.3, write down the sequence of the first three messenger RNA codons from left to right for this portion of the protein. So we're looking at alanine, histidine, and at least tryptophan. So we go to alanine, and alanine was, look, GCU. So we've already got it.
Yes.
Okay, and then hist, we'll have to go up just a touch. And hist is CAU. And tryptophan is going to be UGG. And for that, they give you 3 marks. How easy is that, hey?
Yes.
Okay, now, the following is a sequence of base triplets in DNA. Now, people, look here. This is DNA. So they're giving you what is on the DNA, and they say, if guanine found in the first base triplet, so this one here, is removed, explain how this would affect the structure of the protein. So we change this guanine here. How is that going to affect the structure of this protein that we're putting together? Because remember, this part here is a piece. This is a piece of the protein. So they tell us of the protein molecule. Now we change this G here. How is that going to affect the protein? The protein is going to be changed. So the protein will be altered. Or you could say the protein would be changed and result in a different.
Okay, I was struggling with that question.
Sorry?
Is the question that I was struggling with.
Is this the one you're struggling with?
Yes.
Okay, look. Okay, let's do this. Remember?
But now I can understand.
Okay, if I have adenine, adenine is always going to bond with thymine. Okay, and guanine will always bond with cytosine. Adenine and guanine are your purines, and thymine and cytosine are your pyrimidines. Okay, so these have always got a Y in them. So there's a Y there and there's a Y there. There's thymine and cytosine and the Y for pyrimidines. That's how you remember that. But they always bond like this. That's to make up DNA. When we have RNA, we're going to have adenine will always bond to uracil because there is no thymine in RNA. And guanine will always bond to cytosine just like it does for DNA. All right. So if you remember that, that's fine. Now, if I have a triplet, a base triplet, triplet tells you that there are 3 pieces. And it comes from the nitrogenous bases. So let's say I have adenine and guanine and cytosine. That is my little triplet base. It's all my base triplet. There are 3 parts to it, 3 nitrogenous bases, okay? And I change one of these, either the A or the G or the C. So let's say I change the A to G. So now it becomes that. Or I change the A to cytosine. So it becomes CGC. Or I change the A to uracil. It becomes that. Do you follow? The minute I change the triplet, it will therefore alter the amino acid. And if it changes the amino acid, that is going to result in an altered protein. Does that make sense, Postnet?
Yes.
Are you sure, Mahan?
Yes, I'm sure.
Okay. So that's all you have to remember every single time is A to T and G to C. And if I change any of these letters, okay, so if it's changed in the first, this is my first triplet, that's my second one, that's my third one, that's my fourth one. If I change that guanine there, then it means that this whole amino acid is going to change. And if the amino acid changes, the whole protein will change. All right? Now name the process that occurs when the sequence of bases in the DNA changes. Now any changes in the DNA, any changes will result in a mutation. Okay?
Yes.
And you must know that because later on, when you get to diversity, we have a problem with mutations. Okay.
Yes.
All right. Okay, Postnet, we're going to go across to the studio for an ad break, and I'll see you all in about 2 minutes. All right?